Your immune system is fully capable of destroying cancer cells. So why doesn't it? The answer lies in the clever tricks cancer cells use to hide from your immune defenses. They hit the brakes on your T cells using proteins your body was never meant to fight. In this video, we'll break down how immunotherapy works, specifically how checkpoint inhibitors release those brakes, how CAR T cells and bispecific antibodies offer alternative strategies, and what's new in this rapidly evolving field. Get ready to explore the science behind one of medicine's most exciting frontiers. Decades cancer treatment meant surgery, radiation, and chemotherapy, but now immunotherapy has emerged as the fourth pillar. Unlike traditional treatments that attack cancer cells directly, immunotherapy activates the patient's own immune system to seek out and destroy cancer. This approach includes immune checkpoint inhibitors, CAR T-cell therapy, bispecific antibodies, and monoclonal antibodies. Together, these therapies have become one of the fastest-growing areas in oncology drug development, offering new hope and options for patients worldwide. Immune checkpoints are proteins found on immune cells, especially T cells, that act as off switches. Normally, these checkpoints protect healthy tissue by preventing the immune system from attacking the body's own cells. However, cancer cells can hijack these off switches, activating checkpoint proteins to convince the immune system to leave them alone. Immune checkpoint inhibitors are drugs designed to block this interaction, releasing the brakes and allowing T cells to recognize and attack cancer cells again. This concept has revolutionized cancer therapy by harnessing the body's own defenses. PD-1, or programmed death receptor 1, is a checkpoint receptor mainly found on T cells. Its partner, PD-L1, or programmed death ligand 1, is present on many cell types, including a large number of cancer cells. In a healthy immune system, when PD-1 on a T cell binds to PD-L1, it sends an inhibitory signal that calms the T cell down. This mechanism is essential for preventing autoimmune disease, ensuring the immune system doesn't attack healthy tissue. But cancer cells can exploit this pathway, making it a critical target for new therapies. Cancer cells often increase the amount of PD-L1 on their surface, essentially disguising themselves with the body's own safety mechanism. When T cells infiltrate a tumor and encounter high PD-L1 expression, they receive a strong inhibitory signal. This leads to a state called T cell exhaustion, where T cells lose their ability to multiply, release cytokines, and kill cancer cells, even when they're right next to the tumor. This clever evasion tactic is one reason why tumors can grow unchecked, and why targeting this pathway is so important. Immune checkpoint inhibitors targeting the PD-1, PD-L1 pathway fall into two main categories. PD-1 inhibitors like nivolumab and pembrolizumab bind directly to PD-1 on T cells, preventing PD-L1 from attaching. PD-L1 inhibitors such as atezolizumab, durvalumab, and avelumab bind to PD-L1 on tumor cells, blocking the interaction from the other side. Either way, the inhibitory signal is interrupted, T cells regain their function, and the immune system can launch a real attack on the tumor. These drugs have transformed outcomes in cancers like melanoma, lung, and kidney cancer. CTLA-4 is another checkpoint protein found on T cells, but it works earlier in the activation process than PD-1. For a T cell to fully activate, it needs two signals, one from the T cell receptor recognizing an antigen and a costimulatory signal through the CD28 receptor. CTLA-4 competes with CD28 for binding partners, but binds them with much higher affinity. By winning this competition, CTLA-4 blocks the costimulatory signal, preventing full T cell activation. This mechanism is another way the immune system maintains balance and another target for cancer therapy. Cancer cells can exploit CTLA-4 by increasing its presence on tumor-infiltrating lymphocytes, creating an environment that shields the tumor from immune attack. Ipilimumab, a monoclonal antibody that blocks CTLA-4, was one of the first checkpoint inhibitors approved and remains a key treatment for melanoma. It's often used together with a PD-1 inhibitor for a stronger, combined effect. This dual approach can lead to more robust immune responses, offering hope for patients with difficult-to-treat cancers. While checkpoint inhibitors release the brakes on existing T cells, CAR T-cell therapy takes things a step further by engineering a new weapon. In this approach, a patient's own T cells are collected and genetically modified in the lab to express a chimeric antigen receptor that recognizes a specific target on cancer cells. These engineered T cells are then expanded into the billions and infused back into the patient. CAR T therapy has produced deep, long-lasting remissions in certain blood cancers, with some patients remaining in remission for years. Specific antibodies are a rapidly growing class of immunotherapy. Each molecule is designed to bind two different targets at once, typically grabbing a T cell with one arm and a cancer cell with the other. This physical connection forces the T cell and cancer cell together, allowing the T cell to kill the cancer cell on contact. Tarlatamab, which targets DLL3 in small cell lung cancer, received FDA approval in 2025, marking a major milestone for this approach. Similar therapies are now approved for multiple myeloma and lymphoma. Research in immunotherapy is moving beyond CAR T cells. Tumor-infiltrating lymphocyte TIL therapy takes T cells directly from a patient's tumor, expands them in the lab, and reinfuses them. This approach recently received FDA approval for metastatic melanoma. Natural killer NK cell therapies, including CAR NK cells, are also being explored as potentially less toxic alternatives. Researchers are now combining CAR T cells with checkpoint inhibitors, oncolytic viruses, and cancer vaccines to make these treatments even more powerful and harder for cancer cells to escape. Releasing the brakes on the immune system isn't without risks. Checkpoint inhibitors can reduce the body's ability to distinguish self from non-self, sometimes triggering immune-related adverse events. This means the activated immune system may attack healthy tissues, affecting the skin, gut, lungs, endocrine glands, and occasionally the heart. Early recognition and management of these reactions, often with steroids, is now a critical part of patient care. Counseling patients about these risks is an essential responsibility for the entire oncology team, including pharmacists. Checkpoint inhibitors to CAR T cells and bispecific antibodies, immunotherapy has fundamentally changed what's possible in oncology. Today, these therapies account for the majority of new cancer drug approvals, reflecting their transformative impact. As research continues, we can expect even more innovative treatments that harness the power of the immune system. The future of cancer therapy is immune, and ongoing advances promise new hope for patients everywhere. Let's take a step back and look at the immune system itself. It's a complex network of cells, tissues, and organs working together to defend the body against harmful invaders like bacteria, viruses, and even cancer cells. T cells are a crucial part of this defense, patrolling the body and identifying threats. However, the immune system must also avoid attacking healthy cells, which is where checkpoints come into play. Understanding this balance is key to appreciating how immunotherapy works. E cells are specialized white blood cells that play a central role in the immune response. They can recognize and destroy infected or abnormal cells, including cancer cells. However, their activity is tightly regulated by checkpoint proteins to prevent unwanted damage to healthy tissues. When these regulatory mechanisms are hijacked by cancer, T cells become less effective, allowing tumors to grow. This is why therapies that reactivate T cells are so promising in cancer treatment. Immune checkpoints are essential for preventing autoimmunity, a condition where the immune system attacks the body's own tissues. By acting as off switches, checkpoints like PD-1 and CTLA-4 ensure that T cells don't mistakenly target healthy cells. This balance is delicate. Too much inhibition allows cancer to grow, while too little can lead to autoimmune diseases. Immunotherapy aims to tip the balance just enough to fight cancer without causing excessive harm. Tumor microenvironment is the ecosystem surrounding a tumor, including immune cells, blood vessels, and signaling molecules. Cancer cells can manipulate this environment to suppress immune responses, often by increasing PD-L1 expression. This creates a protective shield that makes it even harder for T cells to attack. Understanding the tumor microenvironment is crucial for developing effective immunotherapies that can break through these defenses. E-cell exhaustion is a state where T cells lose their ability to function effectively after prolonged exposure to cancer or chronic infection. They stop proliferating, release fewer cytokines, and can't kill cancer cells efficiently. This exhaustion is driven by persistent inhibitory signals, like those from PD-1 and PD-L1. Revering T-cell exhaustion is a major goal of immunotherapy, aiming to restore the immune system's ability to fight cancer. Checkpoint inhibitors have revolutionized cancer treatment by unleashing the power of the immune system. By blocking proteins like PD-1, PD-L1, and CTLA-4, these drugs allow T cells to attack cancer cells more effectively. The results have been dramatic in some cancers, with long-term remissions and improved survival rates. However, not all patients respond, and researchers are working to understand why and how to expand the benefits to more people. PD-1 inhibitors, such as nivolumab and pembrolizumab, work by binding directly to the PD-1 receptor on T cells. This prevents PD-L1 from attaching and sending its inhibitory signal. As a result, T cells can remain active and attack cancer cells. These drugs have shown remarkable success in treating cancers like melanoma, non-small cell lung cancer, and kidney cancer, offering new hope for patients who previously had few options. PD-L1 inhibitors, including atezolizumab, durvalumab, and avelumab, take a different approach by binding to PD-L1 on tumor cells. This blocks the interaction with PD-1 from the tumor side, preventing the inhibitory signal. The end result is the same: T cells are reactivated and can target cancer cells. These drugs have expanded the range of cancers that can be treated with immunotherapy, making this approach more widely available. Combining different immunotherapies, such as PD-1 and CTLA-4 inhibitors, can produce stronger and more durable responses. This approach takes advantage of different mechanisms to overcome cancer's defenses. For example, using ipilimumab with a PD-1 inhibitor has become a standard treatment for advanced melanoma. Combination strategies are also being explored in other cancers, aiming to improve outcomes for more patients. Monoclonal antibodies are lab-made proteins that can target specific molecules on cancer cells or immune cells. They form the backbone of many immunotherapies, including checkpoint inhibitors and bispecific antibodies. By precisely targeting key pathways, monoclonal antibodies can enhance the immune response against cancer while minimizing damage to healthy tissues. Their versatility makes them a powerful tool in the fight against cancer. The process of creating CAR T cells starts with collecting a patient's T cells through a blood draw. In the lab, these cells are genetically engineered to express a chimeric antigen receptor that recognizes a specific cancer marker. The modified cells are then multiplied into the billions and infused back into the patient. This personalized approach allows the immune system to specifically target and destroy cancer cells, leading to impressive results in certain blood cancers. CAR T-cell therapy has produced deep, long-lasting remissions in patients with certain blood cancers, such as diffuse large B-cell lymphoma. 5-year follow-up data shows that about half of treated patients remain in remission with some approved products. These results are especially encouraging for patients who had exhausted other treatment options, highlighting the potential of engineered immune cells to change the course of disease. Specific antibodies are unique because they can bind two different targets at once. One arm grabs a T cell while the other attaches to a cancer cell. This physical link brings the T cell and cancer cell together, allowing the T cell to kill the cancer cell directly. This approach is especially promising for solid tumors, where traditional immunotherapies have struggled. The approval of tarlatamab for small cell lung cancer marks a significant step forward. Tumor-infiltrating lymphocyte (TIL) therapy involves taking T cells directly from a patient's tumor, expanding them in the lab and reinfusing them. These T cells are already primed to recognize the patient's cancer, making them a powerful tool. TIL therapy recently received FDA approval for metastatic melanoma, offering a new option for patients who may not respond to other treatments. This approach is part of a broader effort to personalize cancer therapy. Natural killer NK cells are another type of immune cell being explored for cancer therapy. Unlike T cells, NK cells can recognize and kill abnormal cells without prior sensitization. CAR NK cells are genetically engineered to enhance their targeting abilities, and early research suggests they may be less toxic than CAR T cells. These therapies are still in development, but represent an exciting new frontier in immunotherapy. Researchers are now combining different immunotherapies to create more powerful treatment regimens. For example, CAR T cells are being used alongside checkpoint inhibitors, oncolytic viruses, and cancer vaccines. These combinations aim to overcome resistance and make it harder for cancer cells to escape. The hope is that by attacking cancer from multiple angles, outcomes will improve for more patients. Oncolytic viruses are engineered viruses that selectively infect and kill cancer cells. As they destroy tumor cells, they also stimulate an immune response, making the tumor more visible to the immune system. Combining oncolytic viruses with other immunotherapies is a promising strategy to boost effectiveness and overcome resistance. This innovative approach is still being studied, but could add another tool to the immunotherapy arsenal. Cancer vaccines are designed to train the immune system to recognize and attack cancer cells. Unlike traditional vaccines that prevent infections, cancer vaccines aim to treat existing disease by boosting the body's natural defenses. These vaccines can be used alone or in combination with other immunotherapies, and research is ongoing to develop more effective options for a variety of cancers. Immune-related adverse events, or AE, are side effects that occur when the immune system attacks healthy tissues. These can affect the skin, gut, lungs, endocrine glands, and even the heart. Recognizing and managing these reactions early is critical to patient safety. Most AE can be controlled with steroids or other immunosuppressive drugs, but ongoing monitoring is essential for anyone receiving immunotherapy. Managing the side effects of immunotherapy is a team effort. Oncologists, nurses, and pharmacists all play a role in monitoring patients and providing support. Education is crucial. Patients need to know what symptoms to watch for and when to seek help. With proper management, most side effects can be controlled, allowing patients to continue benefiting from these powerful treatments. Pharmacists are essential members of the oncology team, helping to counsel patients about the risks and benefits of immunotherapy. They assist in monitoring for side effects, managing medications, and ensuring safe and effective treatment. Their expertise supports both patients and healthcare providers, making them a key part of the immunotherapy journey. Not all patients respond to immunotherapy, and researchers are working to identify who will benefit most. Factors like tumor type, genetic mutations, and the presence of certain immune markers can influence response. Personalized medicine aims to match the right treatment to the right patient, maximizing the chances of success while minimizing unnecessary side effects. Biomarkers are measurable indicators that can predict how a patient will respond to immunotherapy. Examples include PD-L1 expression, tumor mutational burden, and specific genetic mutations. Testing for these biomarkers helps doctors select the most effective treatment and avoid unnecessary side effects. As research advances, new biomarkers are being discovered, making immunotherapy more precise and personalized. Some cancers develop resistance to immunotherapy, making treatment less effective over time. Researchers are studying the mechanisms behind resistance, such as changes in the tumor microenvironment or loss of immune markers. By understanding these processes, new strategies can be developed to overcome resistance and restore the effectiveness of immunotherapy. While immunotherapy has shown great success in blood cancers, treating solid tumors presents unique challenges. The tumor microenvironment can be more suppressive, and physical barriers may prevent immune cells from reaching cancer cells. New approaches, such as bispecific antibodies and combination therapies, are being developed to improve outcomes for patients with solid tumors. Immunotherapy has been especially effective in treating blood cancers like lymphoma and leukemia. CAR T-cell therapy and bispecific antibodies have produced deep remissions, even in patients who have failed other treatments. These successes highlight the potential of harnessing the immune system to fight cancer and inspire ongoing research to expand these benefits to more patients. The field of immunotherapy is advancing at a remarkable pace. New drugs, combination strategies, and cellular therapies are being developed and approved every year. This rapid innovation is driven by a deeper understanding of the immune system and cancer biology, as well as advances in biotechnology. The future holds even more promise for patients and clinicians alike. Personalized medicine is transforming cancer care by tailoring treatments to each patient's unique biology. Immunotherapy fits perfectly into this approach, as biomarkers and genetic testing guide therapy selection. This ensures that patients receive the most effective and least toxic treatments, maximizing the benefits of immunotherapy while minimizing risks. Clinical trials are essential for advancing immunotherapy. They test new drugs, combinations, and strategies to determine safety and effectiveness. Patients who participate in clinical trials often gain access to cutting-edge treatments and help shape the future of cancer care. Ongoing research ensures that immunotherapy continues to evolve and improve. Immunotherapy is making a global impact, with new treatments being approved and adopted around the world. Access to these therapies is expanding, and international collaborations are accelerating research. As more countries invest in immunotherapy, the benefits are reaching a wider range of patients, transforming cancer care on a global scale. Future of immunotherapy is bright, with new therapies and technologies on the horizon. Advances in gene editing, synthetic biology, and artificial intelligence are opening up new possibilities for cancer treatment. As our understanding of the immune system deepens, we can expect even more effective and personalized therapies that offer hope to patients everywhere. Immunotherapy has ushered in a new era in cancer treatment, transforming the outlook for many patients. By harnessing the power of the immune system, therapies like checkpoint inhibitors, CAR T cells, and bispecific antibodies are changing what's possible in oncology. As research continues, the promise of even more effective and personalized treatments grows. The immune revolution is here, and it's just getting started.